Acetyl-CoA, yes. Citrate, yes. L and M stands for liver versus muscle, which I'll get to in a moment. So ATP inhibits, acetyl-CoA inhibits, fatty acids inhibit, right? So here, these three refer to this over here. Fructose 1,6-bisphosphate actually activates it. Okay? If there's a lot of that around, the idea is that we have the capacity to run more glycolysis. So maybe we should be doing a feed forward, right? That's going to be played off against how much of this stuff there is that's going to actually block it. Alanine is a transamination product of pyruvate. Right? Substitute NH3+ and put in substitute those for C double bond O. We call the transamination reaction. So that'll feed back. So that's related to it. So if there's a lot of pyruvate, there's a lot of alanine. So that's like, one step removed from product inhibition. Okay. So those are energy state of the cell or product inhibition types of allosteric feedbacks.
So is there any relation between the fact that there's two allosteric regulators and one activator? So does that kind of like mean that if...
This is actually showing 4 inhibitors and 1 activator.
Oh, yeah. In that case, so does that mean that you can ramp it down more slowly and controlled, but when you need it to activate, you can just do it much quicker than you want?
It's hard to go from the number of possible activators and inhibitors to the degree of control that you ultimately have as a consequence of that combination. Like, I wouldn't say that because there's only one feed-forward activator that means it's necessarily less effective in activation, or it never gets that far. So then we have hormonal control, right? So glucagon is a pancreatic hormone. Okay, so different organs in the body, like pituitary glands and the pancreas, secrete hormones. One class of hormone is short peptides. 29 amino acids is somewhere between a short peptide and a real protein. Certainly not a protein because it has no interior core. But it has determinants that can be recognized. So glucagon is released in the pancreas when there's a low blood sugar situation in the blood. Okay. Which means that you want to be releasing glucose. Right? So what does glucagon do? It activates an enzyme called protein kinase, cyclic AMP dependent protein kinase. You recall what cyclic AMP looks like. It's just a metabolite with a cycle around the ATP phosphate, a single phosphate. And what that kinase does is it phosphorylates pyruvate kinase. When pyruvate kinase is phosphorylated, it is inactive. And notice here it says L. Right? So L means liver. Right? So we're talking about the liver enzyme here because the liver has the metabolic role. So you want to turn off glycolysis when there's low blood sugar, right? Because you'll be here. Right? pKa, glucagon, there'll be some pathway of transduction in ways that you studied at least partially last term, and I will show you some slides again on Friday. Activates this enzyme, which uses ATP to transfer phosphate onto the glycolytic enzyme, and in this state, it's inactive. So maybe the phosphate is like right in its active site, can't find PEP anymore, or whatever, something like that, right? And then there's a phosphatase around that can also get triggered, and the phosphatase gets triggered when the glucagon concentration becomes low. That's not really depicted on here, but PP means protein phosphatase. Right? So that's a way of taking the phosphate back off. Right? So, and then if glucagon is in a low concentration, meaning you don't have a low blood sugar situation. Okay, so this is just to show you that part of that regulation, and I think this is a slide you probably saw last term, but here we are in a liver cell. Here's the hormone. Right? There's a G protein-based mechanism in getting in the signaling, in the membrane-based signaling, right, which results in the activation of adenylyl cyclase to make cyclic AMP. Cyclic AMP activates pKa, and this pKa is then phosphorylating pyruvate kinase. Right? So this is the connection between that pKa molecule and the glucagon in a little bit more detail, going through these membrane-bound G proteins and the activation first of this other enzyme, adenylyl cyclase. And then there's more to this pathway, but it involves phosphorylase kinase, glycogen phosphorylase, glycogen, so you can see that there will be effects later on the glycogen metabolism, so we'll come back to that on Friday. Fates of pyruvate. We know that pyruvate is a central metabolite because you can go forward to acetyl-CoA, or backwards to oxaloacetate, and then up to gluconeogenesis. Right? So acetyl-CoA is a key activator and inhibitor here. Right? So we've got two enzymes. So the two main enzymes that operate on pyruvate in the mitochondria are PDC and pyruvate carboxylase to take you in the forward direction for energy or the reverse reaction. Right, so this is after pyruvate's been translocated. So we're post-glycolysis here, but we're regulating what happens to pyruvate later. Now there's a bunch of other regulation, which we're actually skipping this year because of the snow day thing, but there's regulation on citric acid cycle enzymes as well. The principles are pretty much the same as we were already getting, which is why I thought it would that part. But this gives you a sense that, this is going into the TCA cycle. We've seen regulation here, now we get regulation here. Is there going to be regulation here too? The answer is yes. And the principles of it are, analogous to what we're already talking about. So, what about this further level of control on PFK with a different metabolite called fructose 2,6-bisphosphate? So here's PFK1, here's fructose bisphosphatase 1. These are the allosteric effectors that we talked about in the context of PFK1, where the low energy ones activated and the high energy ones inhibited. Here's AMP is actually working on both of them. So that's that's a neat case of the same effector either activating one side and inhibiting the other side. So that's a really effective way of making sure one enzyme is turned off and the other one's turned on. Right? And recall that AMP is uniquely suited to do that because small changes in its concentration have large percentage effects on the concentration of the cell. So small changes in absolute concentration of AMP, because its level is low in the cell, have large percentage effects on how much there is. Right? And so that it moves you up and down the concentration curve much more strongly. Okay. So this additional level of regulatory control involves this F26BP. And so how does F26BP work? It works to activate PFK1. In fact, it was on the earlier slide. We just kind of showed there's F26BP. So if you have it, it works very well. If you don't have it, it works very badly. This is the concentration of the other substrate, and you see it stays very low. So this is actually helping address the issue of how you keep it completely turned off when you want it turned off. Right? Just don't have any of this guy around. So here's the other side of it, the gluconeogenesis enzyme. In this case, if we don't have it, right, the absence of it means it works. If you have it, it's a big inhibitor. Right? So this metabolite inhibits one side and does not inhibit the other side, and that's embodied here. So it inhibits gluconeogenesis, it activates glycolysis. Right? This is what it looks like. It's just like fructose 1,6 except fructose 2,6. So then the question is, well, where does it come from? So